Hey, can you hear me?
Hey, yeah, sorry, I was having some issues with the audio.
Yeah, no, I could tell, so I didn't want to assume anything. So it seems like you're going to be potentially the only person here. So um, if you want, we can get started right now because I'm not sure if anybody else is coming.
Yeah, sure.
Okay. All right. So I think I'm recording. Thank you for making it anyways, because I know it's in the middle of summer. Probably have a lot of things to do, so I'm glad you could spend some time with us here at 10 a.m.
Yeah,
in the
no,
morning.
no problem. Thanks for doing this.
Yeah, yeah, yeah. All right. So um, let me share my screen first. Um, I did not have this prepared. All right, so we are going to be doing, and I'm sure you've been to previous sessions with us, so um, you probably know how this goes. But we'll be doing um, kind of a walkthrough of a just like a PowerPoint that I made and covering some concepts, and then we'll go into some Jack Westin questions in a practice passage that will kind of cover the same concepts and make sure you really understand and you like walk away with something that you learn. Um, so we can definitely get started. Um, File. How do you even present this? Very interesting. View. There we go. All right. So icebreaker. Kind of an awkward icebreaker here, but I guess we can share with each other what our favorite food is. Mine is spaghetti.
Um, I love spaghetti too, but I guess my favorite food is chalupas from Taco Bell.
Oh wow, that's very interesting. I
Yeah.
don't usually go to Taco Bell, so I'm not too sure what that is, but I'm sure it tastes great.
Yeah,
Uh,
it's just a soft taco. So,
just a soft taco. Okay, yeah, that
yeah.
sounds nice. Sounds nice. Okay, very nice. Very quick icebreaker. All right, so what we're doing today is kind of a biochem passage. It's going to be a lot about metabolism, a lot about certain enzymes involved in fatty acid synthesis, breakdown, and stuff like that. So I wanted to start off with like some chemistry to just know what we're talking about when we get to the passage. So some of this, I guess most of this should be review, but we have on the left here, a diagram showing oleic acid, linoleic acid, palmitic acid. You don't really need to know like specifically oleic acid is a monounsaturated fatty acid or that linoleic acid is polyunsaturated, but just be able to recognize them from the chemical structure. Monounsaturated, right? Mono is means one, so monounsaturated means it has one double bond. Right? So polyunsaturated fatty acid will have two double bonds, or it could have three or four, but it just has to have more than one. Um, Palmitic acid is a saturated fatty acid, so it has no double bonds, right? So all single bonds. And remember here, the C double bonded to the O doesn't count as a double bond. That's just part of the carboxylic acid group, which all the acids have. Right, so on the right, I essentially say the same thing. And just like kind of as a quick concept check, and trying to really apply this to questions that the MCAT might throw at you. So they might ask, for example, given that you have three fatty acids, one of them is monounsaturated, one of them is polyunsaturated, and one of them is saturated, and they're around the same number of carbons long, which is important because the only difference would be the number of double bonds. Then which of the three would have the highest melting point first?
So I think like so this is kind of tricky for me. I don't know
Sure,
because
sure. That's good.
if
That's
it
good.
has the highest melting point,
Hmm.
that indicates that is it like it's right now would be a solid right now, and what would you do to melt it?
Yeah, you can think of it like that. Um, if it helps, well, let me tell you how I usually think of, and yeah, it can be kind of complicated, highest melting point, boiling point, stuff like that. I would just say, um, don't focus on the melting point part. Just focus on which one would take the most energy to change phases. So whether it's from a solid to a liquid, like in melting, or liquid to a gas, boiling, usually the one with the highest melting point will also have the highest boiling point. So really focus not only on melting point, but just like which one takes the most energy to pull the molecules apart? Which one has the most attractive forces? If that makes sense.
Got it. Probably the hmm, maybe I'm thinking too hard, but like
Just give
I
it a
know
go.
like in I don't know in phospholipids like usually one of the fatty acids is bent and one of the
Yeah,
fatty acids is straight saturated,
yeah.
which gives the membrane its fluidity.
Mm-hmm.
So I'm thinking like the one at the highest melting point would be the one that's the least fluid right now. If you
Okay.
think about it, it's either that or simpler like okay, the one with the most double bonds
Hmm.
would be the highest one. So probably linoleic acid.
Okay. So let me backtrack a little bit. And there was one statement that you said which was correct, which is the one that was least fluid would have the highest melting point, right? And also the reason you said like the reason that you gave was also correct because if it's less fluid, that kind of means that it can't, brush past each other. The molecules can't go past each other very easily, and they're kind of just tightly packed together, right? And whenever molecules are tightly packed together, that lets the attractive forces between them to be a lot stronger because they're all very close to each other, and the closer you are, the more attraction there is. So because of that, there's more attractive forces. It's harder to pull them apart, so you have to put in more energy, and it has a higher melting point and higher boiling point. So of the three, we have to look for which one is the least fluid. And to give you a hint, it all comes down to the shape. So which of the three would have the best shape for sticking together and being not fluid?
Palmitic acid.
Exactly, right? Because it's just like a really long straight chain. Um, Whereas oleic acid and linoleic acid, you're gonna have bends and kinks. Those are probably words that you remember just because we have double bonds. So it's gonna be bent, as you said, like the phospholipid. So it just can't pack together as closely. Um, yeah, so because they're not close enough together, it's a lot easier for them to break apart and change phases. So, going along with that for number two, which one has the lowest melting point? It's probably going to be the one with the polyunsaturated fatty acid, the one with more than one double bond. Um, and while we're actually on this topic, I should probably go over the difference between cis and trans double bonds. Um, which one between cis and trans would lead to a higher melting point? It might take some time to think about that.
Probably trans.
Trans. Okay, so before I answer your question, yeah, I believe you're correct. Right. So if we draw this out, and this is probably something you probably mentally envisioned in your head, this would be cis or trans.
That would be cis.
That would be cis, right? So, um, and the way cis works is like because you have something like this, right? There is kind of a bend here because going in this direction at first, and then because of the cis, it kind of bounces off kind of, and there's a kink. So because of that, cis fatty acids will take up a lot more space, whereas trans, the way you kind of see it written is more like this. So um, it's kind of similar to something that is um, saturated or doesn't have a double bond, but that would be why trans would have a higher melting point. And kind of an interesting point, like that's why you always see animal fats, or yeah, that's why you always see animal fats, right, as having trans fats, which are supposedly or not supposedly, but they are very bad for you. And the fats are almost always very bad for you are going to be more animal fats and not really vegetable oils or vegetable fats because those are more likely to be cooking oils, right? Stuff that's liquid at room temperature. Well, the animal fat will be solid at room temperature because it has that higher melting point. Um, just relating all of those things together. And I guess that also kind of relates to, questions 3 and 4. So which one would be most likely to be solid and which one would be most likely to be liquid?
The saturated would be solid,
Hmm.
and the liquid would be the linoleic and oleic acids.
Exactly, right?
More
And
linoleic. Yeah.
yeah, exactly. So yeah, so I mean, yeah, right? So just the lower the melting point, it's more likely going to be liquid. The higher the melting point, it's more likely to be solid. And I just find it interesting. You can kind of relate it to vegetable versus animal sourced fats. So hopefully that makes sense. Do you have any questions on what we just covered?
No, I think I'm good.
All right, sounds good. All right, so kind of moving on to how do we actually create um, this kind of fatty acids in our body? There are two main pathways that we use to create triacylglycerols. Um, okay, if you can see my mouse here. So triacylglycerols are what's the other name for triacylglycerol? Like what do we commonly know it as?
Lipids.
Lipids, yeah, just fats. Fatty acids. It was kind of a trick question, but yeah, fatty acids. Stuff that we can break down and use for energy, right? So if you go back to like metabolism, right, we like to use carbohydrates or sugars, glucose, which is what we eat. Um, but if we have stuff left over after a meal, then we can put it into glycogen, which is kind of the long-term storage form of glucose, or even going more long-term, we can put them into fats and store them. And that kind of method, or not like the way we store them is through triacylglycerols, is we store these fatty acids in our fat cells. Um, ooh, sorry. Did I? Okay. Um, Okay, so there are two main pathways to make these triacylglycerols: the monoacylglycerol pathway and the glycerol phosphate pathway. Um, and as you can see here on the right, I wouldn't focus too much on like the specific enzymes and molecules, but you should know the ones in red. Now the starting one is obviously monoacylglycerol. The starting one for the glycerol phosphate pathway is G3P, which is glycerol 3-phosphate. Um, I don't know if you've taken biochemistry, but do you know of like a specific reaction or pathway where you've seen G3P before?
Um, Glucose.
Glucose, so which reaction that uses glucose also kind of creates G3P along the way?
Glycolysis.
Exactly, right? So glycolysis, one of the intermediates is actually G3P. So we can actually kind of pull from that and use that same G3P and maybe divert it into another pathway and use it for triacylglycerol synthesis. So this G3P can go through some steps. And notice that both pathways make diacylglycerol. So this is kind of, the main intermediate that kind of connects both pathways. And eventually you can add an acyl group, right? Because this has two acyl groups, diacylglycerol. So you add another acyl group and you make triacylglycerol, which is the fat that we want. All right. Okay, so this is kind of a broad overview of where do these processes actually occur in our body, right? Where does this all take place? So it's actually kind of good to just go through all of this together. Dietary carbohydrates, I apologize for these misspellings. Dietary carbohydrates and dietary proteins, so stuff that we eat from our food, they can be broken down into acetyl-CoA, right? That was the same acetyl-CoA that we make from glycolysis and the citric acid cycle. So acetyl-CoA, you'll see it a lot. It's just a way that we can break it down and then further break it down to make energy. So this acetyl-CoA can go through the citric acid cycle, and here on this arrow kind of also shows, the electron transport chain, oxidative phosphorylation, all that good stuff to make ATP. So from here to here is kind of just glycolysis and normal breakdown of glucose. But we can also not go through the citric acid cycle, but instead use the acetyl-CoA to make lipids through lipogenesis. Right, so lipids is actually kind of a broad term that includes fatty acids, waxes, steroids, stuff like that. So you can create a whole lot of different lipid molecules, and some of this includes fatty acids, and you can combine glycerol and fatty acids to make triglycerides, right? Because we learned that one glycerol plus 3 fatty acids makes 1 triglyceride, right? Because the glycerol has kind of like 3 prongs to it, and 3 fatty acids can come attached to it. So once you make the triglycerides, and this is all occurring in the liver, right? So, and you can see here on the right, lipogenesis, which is making fats, occurs in liver cells and fat cells. So these are the two main areas where this process of making triglycerides is going to take place. Then once you make those fats, you're going to transport them into the blood and then going to these fat cells, right? Because that's where we store the fat, and that's where lipid storage happens. So another name for fat cells is going to be adipocytes, or adipocytes. I never know how to pronounce it. And so once it's going to be stored there, then when you are maybe starving, you're not in a good spot, your body needs that energy from the fats. It's going to start breaking down the fats, and that's called lipolysis. And again, that also occurs in adipocytes because that's where you're storing the fats. So you're just going to take them out and break them down again, and release the energy. So I like this. I kind of highlighted this in red here because I kind of like this diagram. It shows you that like the liver is kind of at the center of everything. And I have here like an overgeneralization, but something that could be useful in case you have to guess on a question is like whenever you're talking about where some metabolic process occurs, more often than not, it's going to be in the liver, or have to do with the liver because the liver controls a lot of glucose, fatty acid metabolism. You'll learn later like ketogenesis also occurs in the liver. So it does a lot of stuff with handling the energy needs of our body. And then from the liver, it connects to the brain because the brain needs glucose, it needs the energy. It always, it's one of the most energy intensive organs. So it always needs energy to survive. And then muscle and adipose tissue. So it kind of just relates everything together. All right. Any questions on that?
No, I think I'm good.
All right. So last but not least, or actually, no, I think we have one more slide. So main biochem reaction types you should know for the MCAT. So just, a quick overview of, okay, we have hydration here, ligation, isomerization, stuff like that. Hydration, so it's always easy to mix up hydration and dehydration, like condensation stuff. The main way I like to remember it is just like kind of like look into the words. So hydration is like when you're hydrated, you're adding water to your body, right? So hydration always involves adding the water. And when we add water, just associate adding water with breaking down stuff because, I don't know, water just likes to break down molecules. So here we have A, which is like a bigger molecule. Once you add the water, it's going to break down into B and C, which are smaller molecules. We also have ligation, right? Ligation is when you're ligating something, you're tying them together. So A plus B becomes AB. So you'll see this in like, DNA, right? Because when we talk about DNA ligase or, just adding or, connecting two big molecules together to make an even bigger molecule, that's ligation. Isomerization, right? A to B, make an isomer of it. So very similar, but just, a different shape, a different form. Oxidation reduction, right? So A plus B with 2 electrons becomes A with 2 electrons plus B. So you're just moving the electrons from one thing to another. Dehydration. So dehydration, right? When you're dehydrated, your body's losing water. So the water is going to be on the product side, and it's going to be the opposite of hydration. So if hydration was breaking stuff down, dehydration is going to be building bigger stuff. So A plus B going to AB. Saponification is triglyceride plus base goes to glycerol plus fatty acid salts. So 6 and 7 kind of have to do with more fatty acid specific stuff. Saponification, so that is making soaps, right? And really what soaps are fatty acid salts. So we're breaking down fats or lipids or triglycerides using base to create glycerol and fatty acids. And we actually use this to make the salts that we use in our household products. Esterification is the kind of the opposite of this. You're adding the carboxylic acid plus alcohol to make ester and water. So it's kind of hard to see, but like saponification and esterification are actually opposites of each other because this glycerol is like this alcohol, right? They both end in OL. Glycerol is actually just it's a molecule with 3 alcohol groups. And then these fatty acid salts are just the same thing as a carboxylic acid. And you're making an ester plus water. So the ester here is just the triglyceride, right? Because remember, triglycerides are esters. That's what their functional group is. So the reason I present all of this is, first of all, you basically should memorize or at least understand in a lot of detail all of these reactions for the MCAT, and also the passage that we're going to be discussing will kind of rely on 6th and 7th. It's good to know them. Any questions?
No, I'm good.
All right. Okay, so my last slide, and probably one of the most important ones in today's session, is that metabolism reactions come in pairs. So this was something that kind of helped me throughout all sections of the MCAT, or at least all the bio and chem sections, because it's just really key to having a bigger picture, or to being able to understand a bigger picture of metabolism, is to know that for every single reaction, there's something that does the complete opposite, and oftentimes, right, they're going to work in synergy, right? So each one is kind of going to balance the other one out. So for example, glycolysis, gluconeogenesis, glycolysis here. So I listed all the ones that are breaking down stuff right here, and then all the ones that are building stuff up are right here. Let me make sure you can see my mouse. Actually, I can't, not sure. Yeah, okay. So for example, glycolysis is breaking down glucose. Gluconeogenesis is building up glucose from smaller stuff. Glycogenolysis, we're breaking down glycogen to make glucose. Glycogenesis is we're using glucose to make glycogen. So a lot of GL words, but if you really know these down very well and understand the difference, then you should do a lot. It should help you a lot on the exam. And really, I think a lot of it is just looking at the word itself. So whenever you see genesis, you're making something, and whenever you see lysis, you're breaking something down. Yeah. So lipolysis, you're breaking down lipids to make, maybe fatty acids and glycerol. Lipogenesis, you're making lipids from scratch. Ketolysis, breaking down ketone bodies, or breaking down ketone bodies, and then ketogenesis is you're making ketone bodies. Proteolysis, breaking down proteins, protein synthesis, making proteins. And then you have nucleotide degradation and nucleotide synthesis. These two you don't really have to know in detail, like the specific steps of it. I've never really seen it asked before. But it is important to note that glycolysis, proteolysis, protein synthesis, and nucleotide degradation and synthesis, all those reactions take place in all cells of our body. So these, right, because protein synthesis and protein breakdown, all your cells pretty much make proteins, and sooner or later they're going to have to break it down through whatever process, like ubiquitination or any stuff like that. And then obviously, again, DNA and RNA is everywhere in your cells, so you're going to have to make it and break it down. And right, glycolysis, all your cells need energy to survive. The ones that are not highlighted in gray or shaded in gray, these ones don't necessarily occur in every cell. Usually they would occur in the liver. Some of them occur in the muscles. For example, glycogenolysis, right? Because your muscles can store some glycogen and they can break it down, right? Because of the lysis, they can break it down to make glucose and use that as energy when you're exercising. So that's why sometimes glycogenolysis can occur in your muscles. And then, but the liver actually does a lot of these processes as well. So when in doubt, I would just say pick the liver as your answer. Any specific questions on these?
No, I think I'm good.
All right. So that was the last content stuff to go over. So let's move on to the questions. So if you don't mind me asking, when is your test day, or just 1 month?
September 1st.
September 1st. Nice, nice. So you
Yeah.
have what, 2 whole months, more than 2 months to study. So that's great.
Mm-hmm.
Okay. Unfortunately, I guess right here. Let me just make sure I don't have any of the answer choices highlighted. All right, let me share my screen. Okay, so this is yeah. So this is a passage on the title of the passage is called DGAT Isoenzymes. So from first glance it seems kind of complicated, but hopefully what we just covered kind of make these graphs a little bit more clear. Um, so I usually don't like. I mean, I usually like not a big fan of like people reading just because I know like when I read I just completely miss the point of what I'm reading. So if you want, you can. I can actually. I'll copy and paste the Jack Westin link in the chat. And do you think you would be able to go to the link yourself and like kind of read the passage yourself?
Yeah, sure.
Okay. So just give me a heads up whenever you're done reading and ready to answer the questions.
Okay.
All right.
Okay, yeah, I'm done.
Okay, great. So I'll just go through the passage and kind of show you what I would look out for when I was reading it. Um, right, so triglycerides are neutral lipids of glycerol esterified with three fatty acyl groups. They have multiple functions, right? So serving as a reservoir of fatty acids in membrane biosynthesis. So the major pathway to make triglycerides is the glycerol 3-phosphate pathway, and this uses specific acyl transferases. So here you kind of want to know what acyl transferases are, and this kind of goes back to the figure that we talked about earlier, where we're going from that common intermediate, which was diacylglycerol, in changing that diacylglycerol into a triacylglycerol. So right, if you're going from diacyl to triacyl, you need to add an acyl. So that's what this acyl transferase is doing. It's transferring an acyl group from somewhere else to this compound and giving it that acyl group it needs to become a triacylglycerol. Right, and then the final and only committed step of this pathway is the linkage of a 1,2 diacylglycerol, so that's this, to an acyl CoA, so that's this, and we're linking these two together, catalyzed by this DGAT enzyme, right? And then we're making this triacylglycerol. So as you're reading, kind of want to, go back and forth between this paragraph and this diagram, this figure, and kind of match things and make sure you understand how does this relate and do I really kind of get a gist of what's going on. So then once they know that, researchers wanted to study specificity and sensitivity to a potential inhibitor of DGAT enzymes. So to optimize, whatever detection, they implemented an assay that uses a fluorescent DG substrate. So it's called NBD-DG. So it kind of complicated, but what does this NBD thing stand for, right? So you can see here it's this weird, kind of benzene thing with a heterocyclic ring on the top with this nitrogen dioxide group sticking out. So this NBD thing, the main focus, right, is it's a fluorescent thing. So what I'm looking at here is um, the NBD group is just the fluorescent group that's going to allow you to visualize it. So it's not important for the actual chemistry. It's just like something that we add on to make sure we can see it through a microscope. And the main thing here is this, this actual 1-decanoyl, 2-decanoyl glycerol, which is a diacylglycerol, right? This is the diacylglycerol part. And you can see here, because we have two esters here, that's why we know it's a diacylglycerol. All right, so now we're going to show you the results of their assay, right? So they measured the activity of two DGAT isoenzymes, this and this, using three C18-CoA substrates, C18 colon 0, 18 colon 1, 18 colon 2. So what they're trying to look at here, right? And I always look at the axes first whenever I look at graphs. On the x-axis, we have acyl-CoA, the concentration of acyl-CoA. And then on the y-axis, we have NBDTG. So here we had NBDDG. So this was the fluorescent DG substrate. So it's like you're feeding it in. You're feeding the fluorescent diacylglycerol. And what you're getting out is the NBDTG, which is a fluorescent tag triacylglycerol, right? So it makes sense, right? Because we're feeding in a fluorescently labeled diacylglycerol, we're getting out a fluorescently labeled triacylglycerol, and the fluorescently labeled triacylglycerol is what we have graphed on our graph. So we are comparing this to this, right? So these are the two isoenzymes. So the two versions, basically, of the enzyme, and we're comparing how do these two versions of the enzyme compare to each other. And we have three types of lines, right? C18O, C18-1, C18-2. So, and you've probably definitely seen this before. Whenever you have, for example, number colon number, when it's talking about fatty acids, it's referring to the 18 is the number of carbons, and the 0, 1, or 2 is the number of double bonds. So 18-0 is 18 carbons, 0 double bonds. So that would be saturated, right? And then C18-1 would be monounsaturated, C18-2 would be polyunsaturated. So we're just comparing how does the saturation affect the activity of this enzyme. So that's just the kind of takeaways I would get from this passage. And I like to always read the passage first and get as much out of it as I can, and then look at the questions. Do you have any questions about the passage before we look at the questions?
No, I'm good.
Okay. So question 1, according to figure 3, the preferred substrate for both DGAT isoenzymes is. So I'll give you a few seconds. What do you think is the correct answer here, and why?
I think the correct answer is A.
A. So okay. So yeah, and that I'm pretty sure that's correct, because right, saturated fatty acids, that would be the one with 0 double bonds. And if you look at the graph here, and remember, always just make sure you're actually it would be terrible to know that saturated fatty acids have 0 double bonds, but just make sure like the square, the circle, and the diamond, make sure got those in order. And yeah, so the square obviously is much higher than the other two lines. So because we already said that the axis refers to how efficient the enzyme is, we can be confident saying, because this line is higher, we know the enzyme is making more product, and therefore it's the preferred substrate. All right. I was missing the next button. Okay. Question 2. Researchers hypothesized that vitamin B3, or niacin, inhibits DGAT through a non-competitive mechanism. When incubated in the presence of substrate, enzyme, and inhibitor, which of the following results best supports this hypothesis? All right, so let me know whenever you have your answer ready.
The answer is B.
Yeah, the answer is B, right? Because what was that?
Oh yeah, B. C18O.
Yeah, yeah,
CoA accumulation. Yeah.
exactly, right? So, um, and just kind of going through the question to see why we would pick B, right? Um, so when I read this question, right, I see inhibits DGAT through a non-competitive mechanism. I probably would have highlighted non-competitive too, but going through the question, it doesn't seem that that was actually pretty relevant. So the main thing is, what do we get when we inhibit DGAT? So we're stopping this reaction from going forwards. So whatever is going to accumulate is going to be the stuff. It's going to be the reactants, right? The stuff going into the reaction will start to build up because the reaction is not going forward. So so basically the question is which of these two is being accumulated here? So it is tempting kind of to pick A, right? NBDTG accumulation. Um, but right, the TG refers to the triacylglycerol. So this would be the product. So the product will actually go down in concentration. So we can eliminate A. The C18-0-CoA, right? That's the CoA here. So we know that this is the acyl-CoA that's being used in the reaction. So that's why that would accumulate. NBDDG could be, right? Because this is the diacylglycerol, but then we see D concentration, so that doesn't make sense. And then C18-0 D concentration, it doesn't even have the CoA with it, so I'm not sure where they got that from. All right, great. DGAT enzymes are the most active in which organ?
The liver.
Exactly, right? I kind of really went over this point in the concepts portion, but yeah, the brain, the bone, the kidney. Really the bone, you'll never like that would never really function in metabolism too heavily. Um, brain, the only enzymes that would be most active in the brain would be maybe breaking down ketone bodies, um, but we don't see that here. And right in the kidney, that would be only gluconeogenesis. So right when in doubt, pick the liver. And here DGAT, right, we're making triacylglycerols. That would be lipogenesis. So that definitely occurs in the liver. Another place it would occur in is maybe fat cells, but that also wouldn't be an organ really. If it said maybe most active in which cells, right? And there was no liver, but there was a choice for adipocytes or fat cells, then you would pick that. Okay. I keep missing. Okay. What type of reaction liberates fatty acids from glycerol backbones?
Um, saponification.
Saponification? Yes, that's correct, right? Right, so we are liberating fatty acids from glycerol backbones. So we're basically breaking down the triglyceride, right? Um, so because of that, we're breaking down the triglyceride. We're making soaps. We're making fatty acid solids in glycerols. Esterification would be the opposite. That would be putting together fatty acids with glycerol backbones to make triglycerides. Dehydration would be right losing water. So whenever we're losing water, we are making stuff. So that would again be very similar to esterification, and therefore that would not be right. And ligation, we're not dealing with big molecules or putting them together. So D would not be the correct answer either. All right, so our last question is the products of TGA catabolism do not promote which of the following metabolic pathways?
Gluconeogenesis.
Gluconeogenesis? Hmm. Okay, so how did you come up with gluconeogenesis? Could you explain your reasoning for that?
Yeah, so I know like I just thought like since this pathway makes triacylglycerols, this would be like breaking down glucose to make those fats, but now that I'm saying it out loud, it doesn't make any sense. So,
No worries, no worries. Um,
um,
Before you continue with that line of reasoning, I just want to point out. So it does say TGA catabolism, right? So it's kind of a trick question almost because the whole passage is talking about triglyceride synthesis, but this is saying catabolism. So it's saying the products of breaking down triglycerides. So you kind of have to think about what do we form when we break down triglycerides, right? And then once you go through that, based off the products that we form, where do those products go? Like which pathways do they go into?
glycolysis.
Glycolysis? Okay, so let me ask you this. So what would be the products of TGA catabolism? There's two main ones.
Oh, glycerol and fatty acids.
Exactly, right? So glycerol, was there, okay, so glycerol, so kind of going all the way back to the concepts, there was glycerol 3-phosphate in one of our slides, right? So glycerol kind of feeds in and it creates glycerol 3-phosphate just by adding a phosphate group. So once you get to glycerol 3-phosphate, what pathway did we say that could play a role in?
Um, well, it makes triacylglycerols, but also it would um be a part of glycolysis as well.
Exactly, right? So glycerol 3-phosphate was actually one of the intermediates of glycolysis. So kind of indirectly, right? A lot of things is like it's a lot of application based and some of it is kind of indirect. But because TGA catabolism makes glycerol, that glycerol can become glycerol 3-phosphate, and then that glycerol 3-phosphate can feed into glycolysis. Um, that's why B would not be a correct answer because it does promote glycolysis. And kind of going off of that, if you'll remember, glycolysis and gluconeogenesis, right? They were the ones that go in pairs. They were one of the pairs. So a lot of the reactions in glycolysis are reversible, and when they're reversed, you basically get gluconeogenesis. So it turns out that the reaction that G3P or glycerol 3-phosphate is a part of in glycolysis also occurs in gluconeogenesis. So because G3P is like shared between A and B, A would also not be the correct answer. Yeah, so that's just kind of why gluconeogenesis, glycolysis are actually both incorrect here. C, the Cori cycle. So this one's actually not very much, it's not tested a lot. And you really don't need to know anything specific except like what its main function is. So the Cori cycle is kind of going in between glucose and lactate. So whenever you're exercising, right, and you produce a lot of lactic acid, that's what causes your muscles to be sore. When you produce a lot of lactic acid, obviously you can't just let that build up. So part of breaking it down and redistributing it in the body is through the Cori cycle. That just kind of transfers the lactic acid or the lactate into glucose and back again. So C would actually be the correct answer here because neither glycerol nor fatty acids, which are the products of TGA catabolism, neither of them actually play a role in the Cori cycle. But obviously, you might not be expected to know that. So through the process of elimination, can we eliminate D, which is oxidative phosphorylation? So again, this is also another very indirect one, right? But we have the two products, glycerol and fatty acids. For fatty acids, what can we break down fatty acids into?
Um, what can we break down fatty acids into?
Yes. You may not have covered this yet, so it's okay if you don't know.
Yeah, I don't know.
Yeah, sure. So you'll learn later that fatty acids can actually be broken down into acetyl-CoA. So that's through something called fatty acid oxidation, and that's just the way we break down fats to make energy for us, right? So we can actually break down fatty acids into acetyl-CoA, and that acetyl-CoA can go into stuff like the citric acid cycle, and then D, which is oxidative phosphorylation, right? So just continuing on what acetyl-CoA usually does. And all of that is just to make energy and ATP for our bodies. So because of that, because we can go from triglyceride to glycerol and fatty acids, and from fatty acids to acetyl-CoA, and then acetyl-CoA to citric acid cycle and oxidative phosphorylation, just that whole cycle of steps, D is actually also affected by THA catabolism. So that's why we can eliminate D. So it turns out C is actually the correct answer here. Again, kind of a a higher level thinking question. So definitely a complicated one at that. So do you have any questions about this, or any concerns about why some answers are wrong and C was correct?
No, I think I understand. Yeah.
Okay, cool. Yeah, so this is pretty much it for this passage and pretty much the session. Just like as I just want to get some feedback from you. And actually, I'll stop recording so you can let me know in privacy.